Welcome to the PubMed Daily Review for June 3rd, 2026. Today we scanned 1,657 new open access papers across 399 journals. In this video, we'll cover 10 trending research topics and 10 selected papers for detailed analysis. Trend 1 of 10, specific medical conditions and cases. This trend encompasses detailed reports on particular medical conditions, specific patient cases, and focused treatments or interventions. This cluster covers 196 papers. Representative work includes PX6 aniridia syndrome, la tenectoplace pour la thrombolus d'un AVC ischemique aigu chez un femme de 68 ans, and Avis de Dex pour my 2026. Trend 2 of 10, infectious disease and vaccine research. This trend covers research on infectious disease surveillance, epidemiology, prevention, and treatment, including significant vaccine development efforts. This cluster covers 135 papers. Representative work includes trends in microbiology testing in Australian long-term care facilities, national cohort study, advances in African swine fever vaccine development, challenges and prospects, and preclinical toxicological evaluation of the HIV entry inhibitor peptide 2p23 as a candidate rectovaginal microbicide. Trend 3 of 10, artificial intelligence and machine learning. Exploring advanced AI and machine learning techniques and their practical applications in diverse real-world scenarios. This cluster covers 170 papers. Representative work includes heterogeneity-aware personalized federated learning for household energy forecasting on multi-source real-world smart meter data, DCG, a diversity-enhanced cycle GN for unpaired image-to-image translation, and thermodynamic natural gradient descent, NGDT. Regulates natural gradient steps by a geometric speed cost bound. Trend 4 of 10, public health and health systems. This trend explores health system preparedness, public health interventions, and determinants of health outcomes across diverse populations. This cluster covers 225 papers. Representative work includes Préparer le système de santé canadien aux conflits armes, cadre d'intégration civilo-militaire de services médicaux, effectiveness of the health literacy education program for medical student clerkships, a one-group pre-test post-test study, and determinants of oral pathogen prevalence in northeast Nigeria, a cross-sectional study. Trend 5 of 10, cellular and molecular disease mechanisms. Exploring cellular and molecular mechanisms driving disease pathogenesis, progression, and therapeutic interventions. This cluster covers 162 papers. Representative work includes basic fibroblast growth factor preserves rabbit adipose-derived mesenchymal stromal cells in serum-free culture, regulatory T cells in cancer and inflammation, and insights into the therapeutic strategies for aging and aging-associated diseases. Trend 6 of 10, cancer diagnosis and therapy. Research explores cancer diagnosis methods, therapeutic strategies, and patient care, covering detection, treatment, and side effect management. This cluster covers 159 papers. Representative work includes advanced radiotherapy and systemic therapy in head and neck adenoid cystic carcinoma, current progress and future integrated strategies, unraveling the immune microenvironment in primary CNS lymphoma, and sensitive determination of breast cancer biomarker, CA15-3, using a label-free electrochemical aptasensor based on palladium nanoparticles decorated carbon nanofibers. Trend 7 of 10, materials science and engineering. Exploring material properties, advanced fabrication, and engineering applications across diverse scientific and technological domains. This cluster covers 180 papers. Representative work includes data-driven spatial metamodeling for non-Gaussian digital roughness mapping and precision machining, printable grayscale nanotemplate for full-color organic light-emitting diode near-eye displays with 3D achromatic metalenses, and provenance and economic potential of Egyptian black sands, integrated mineralogical and geochemical insights. Trend 8 of 10, neuroscience and mental health. Exploring neurological underpinnings, biological mechanisms, and research methodologies impacting mental health and related disorders. This cluster covers 137 papers. Representative work includes taking back control of my mind, behavioral characterization of olfactory, auditory, and motor deficits in 5x FAD mice, and shared genetic architecture and pathways linking major depressive disorder and autoimmune thyroid disease. Insights from common and rare variants. Trend 9 of 10, clinical, surgical, and procedural studies. This trend evaluates various clinical, surgical, and procedural interventions and techniques across diverse medical and dental fields. This cluster covers 168 papers. Representative work includes clinical evaluation of cold plasma surface activation using different plasma systems on early implant stability, a randomized controlled within-patient trial, midterm outcomes and outcome predictors after radiofrequency ablation for indeterminate thyroid nodules following functional imaging screening, a retrospective cohort study, and real-time ultrasound-guided versus landmark techniques for thoracic epidural placement in elderly patients, a randomized comparative study. Trend 10 of 10, Agricultural and Environmental Biotechnology. Applying biotechnology to improve agricultural practices, environmental sustainability, and food/health outcomes. This cluster covers 125 papers. Representative work includes meat quality of a slow-growing chicken breed fed soybean meal-free diets, purple sweet potato, Ipomoea batatas L, lamb. Leaves extract prevents weight gain and lipotoxicity in mice fed a high-fat diet by increasing metabolic flexibility and oxidative metabolism in skeletal muscle and brown adipose tissue, and full-length transcriptome resource of the narrow-clawed crayfish. Pontostachys leptodactylus, Esch Scholz, 1832. Up next, our first paper out of 10, AD-GPT, Large Language Models in Alzheimer's Disease by Ziyu Liu and Chao Huang from Department of Statistics, University of Georgia, Athens, Georgia, USA. This paper was selected because it introduces a novel domain-specific large language model, AD-GPT, that addresses the critical challenge of synthesizing vast and complex genomic and clinical data in Alzheimer's disease, significantly advancing. Let's break down its key contributions. Alzheimer's disease research generates an enormous amount of complex data, from genomics to clinical observations. While large language models have shown great promise, general-purpose LLMs often struggle to provide accurate and reliable information in this specialized domain. This creates a critical need for tools that can effectively synthesize and interpret AD-specific knowledge, ensuring researchers have access to trustworthy insights. To address this, our team developed AD-GPT, a specialized framework. We built it upon a foundation of curated genomic resources like GTX and OMIM, combined with a vast collection of over 150,000 AD-related publications from PubMed. The core of AD-GPT is a sophisticated retrieval-augmented generation, or RAG, workflow, featuring a BERT-based query router and fine-tuned llama models, all enhanced with verifiers to ensure accuracy and relevance across tasks, like genetic information retrieval and association study reasoning. When we put AD-GPT to the test, it consistently outperforms strong baseline LLMs across all evaluation metrics. We observed significant improvements in evidence grounding, meaning the information provided was directly supported by our curated data. Crucially, AD-GPT substantially reduced the problem of hallucination, a common challenge with general LLMs, ensuring higher factual consistency and citation validity in its outputs. These results highlight AD-GPT's superior ability to deliver reliable insights. AD-GPT represents a significant step forward, offering a scalable and accurate informatics tool that can truly accelerate Alzheimer's disease research by effectively harmonizing diverse data sources. The innovative RAG workflow, with its task-specific partitioning and verification mechanisms, provides a robust blueprint for developing reliable domain-specific LLMs. Looking ahead, this framework could be expanded to tackle other complex diseases or even integrate real-time clinical data, potentially offering valuable support for diagnostic processes and personalized medicine. Up next, our second paper out of 10, Unveiling the Antibacterial Activity and Mechanisms of BMS 303141 against Staphylococcus aureus by Junhua Ma and Peiyu Li from Department of Infectious Diseases and Shenzhen Key Laboratory for Endogenous Infections, Shenzhen Nanshan People's Hospital, affiliated Nanshan Hospital of Shenzhen University, Shenzhen, China. This paper was selected because it addresses the critical global health crisis of antimicrobial resistance by identifying a novel antibacterial agent, BMS 303141, effective against multi-drug-resistant Staphylococcus aureus, MRSA, through. Let's break down its key contributions. Today, we're diving into a critical area of biomedical research, the fight against antibiotic-resistant bacteria. Staphylococcus aureus, especially its methicillin-resistant form, MRSA, continues to be a major global health threat, making infections incredibly difficult to treat. This urgent need for new solutions drives researchers to discover novel compounds. This paper introduces BMS 303141, a promising new agent, and explores its potential to combat these resistant pathogens. The researchers employed a comprehensive approach to investigate BMS 303141. They started with in vitro studies, determining its minimum inhibitory concentration against various aureus strains, including MRSA, and assessing its ability to inhibit biofilm formation. To understand its mechanism, they utilized advanced techniques like integrative proteomics and whole genome sequencing. Finally, they validated its therapeutic potential in 2 different mouse infection models, simulating real-world infection scenarios. The results were truly exciting. BMS 303141 showed remarkable potency against MRSA, with mic values ranging from 6.25 to 12.5 micromolar. Even more impressively, it effectively inhibited biofilm formation at concentrations below its mic, which is crucial for combating persistent infections. In the animal models, it significantly reduced bacterial burden and accelerated healing, confirming its therapeutic promise. Mechanistically, the compound was found to induce multi-pathway cellular disruption, targeting critical bacterial processes like cell wall integrity and protein synthesis. This study marks a significant step forward in the search for new antibiotics. BMS 303141 stands out as a promising candidate due to its potent activity against multi-drug-resistant aureus and its unique multi-pathway mechanism of action. While these findings are highly encouraging, further preclinical and clinical development will be essential to translate this discovery into a new treatment option for patients. This research truly highlights the potential of targeting novel bacterial pathways to overcome the challenge of antibiotic resistance. Up next, our third paper out of 10. RET receptor tyrosine kinase promotes breast cancer metastasis to the brain, and RET inhibitors pralsetinib and selpercatinib suppress breast cancer brain metastases by Angelina T. Regua and Huiwen Lo from Vivian L. Smith Department of Neurosurgery, McGovern Medical School, the University of Texas Health Science Center at Houston, Texas, USA. This paper was selected because it identifies a novel druggable target, RET receptor tyrosine kinase, for breast cancer brain metastases, BCBM, and demonstrates the efficacy of existing RET inhibitors, offering a highly translational and urgently. Let's break down its key contributions. Breast cancer brain metastases, or BCBM, represent a significant challenge in oncology, leading to very poor outcomes for patients. This is largely because we don't fully understand the biology behind these metastases, and effective treatments that can cross the blood-brain barrier are scarce. This paper aimed to explore the role of a specific protein, RET receptor tyrosine kinase, in driving BCBM and to see if existing RET inhibitors could offer a new therapeutic approach. The researchers employed a multifaceted approach, starting with analyzing human patient samples, specifically 30 BCBM samples compared to primary tumors. They then moved to in vitro studies using brain-tropic breast cancer cell lines, investigating cell viability, apoptosis, and migration. Crucially, they utilized several mouse models, including intracardiac and intracranial injections, to study metastasis and tumor growth in a living system. This comprehensive design allowed them to explore RET's role from human observation to mechanistic validation. The findings were quite compelling. They observed that RET activation was significantly elevated in patient BCBM samples compared to primary tumors, and this high activation was linked to worse brain metastasis-free survival in patients. In their mouse models, overexpressing RET strongly promoted the spread of breast cancer cells specifically to the brain. Most importantly, the RET inhibitors, pralsetinib and selpercatinib, significantly reduced cancer cell viability and migration in lab dishes, and in living mice, they effectively prevented new brain metastases and suppressed the growth of existing brain tumors. This research highlights RET as a crucial new player in breast cancer brain metastasis, offering a fresh target for therapy. The fact that existing FDA-approved RET inhibitors like pralsetinib and selpercatinib, which can cross the blood-brain barrier, showed such strong efficacy in preclinical models is incredibly promising. It suggests these drugs could potentially be repurposed for BCBM patients. Moving forward, clinical trials are essential to confirm these findings and bring this potential treatment closer to patients. Up next, our fourth paper out of 10, Metabolically Engineered Probiotic OMVs as Nanovaccine, Mediating Sequential Immunomodulation for Chronic Bone Infection Immunotherapy by GLA and Xiaobo Feng from Department of Orthopedics, Union Hospital, Tongji Medical College, Huazhong University of Science and Technology, Wuhan, China. This paper was selected because it introduces a highly novel and sophisticated nanovaccine strategy using metabolically engineered probiotic outer membrane vesicles to address the critical and difficult challenge of chronic, drug-resistant bone. Let's break down its key contributions. Chronic bone infections, especially those caused by MRSA, are incredibly challenging to treat. They create an immunosuppressive environment that prevents the body from fighting the infection and healing the bone. This paper explores a novel strategy to tackle this problem by developing a nanovaccine that can sequentially modulate the immune system. Researchers screened Lactococcus lactis OMVs for their ability to activate dendritic cells and repolarize macrophages. They then used metabolic engineering and targeted culture optimization to create NAD+, enriched LAC-OM-versus. This design aimed to achieve a biphasic immunomodulatory effect in models of chronic bone infection. The results were quite impressive. The engineered OMVs achieved remarkable bacterial clearance rates of over 99% against both primary and recurrent MRSA challenges. Crucially, they also led to a tenfold increase in the bone repair rate. This biphasic effect involved early dendritic cell activation and later macrophage repolarization, demonstrating a powerful and targeted immune response. This research offers a groundbreaking approach to treating chronic bone infections by leveraging metabolically engineered probiotics. The ability to sequentially activate antibacterial immunity and then promote bone healing is a significant advancement. While highly promising, future work will focus on translating these findings into clinical applications to benefit patients suffering from these debilitating conditions. Up next, our fifth paper out of 10, metagenomic profiling reveals shared resistance signatures between humans and pigs in Vietnamese smallholder farms by Lu T. Kio Lin and Thierry P. Valaven from Institute of Tropical Medicine, Universitätsklinikum Tübingen and German Center for Infection Research, DZF, Tübingen, Germany. This paper was selected because it provides critical metagenomic evidence of shared antimicrobial resistance gene signatures between humans and livestock in a high-burden region, directly addressing the urgent global health crisis of antimicrobial resistance through a One Health approach. Let's break down its key contributions. Antimicrobial resistance, or AMR, is a critical global health challenge. This study aimed to understand how much antibiotic resistance is shared between humans and livestock, particularly in regions where AMR is a major problem, like Vietnam. Understanding these connections is vital for developing effective strategies to combat the spread of superbugs. Researchers conducted a cross-sectional study in Vietnam, collecting paired fecal samples from both pigs and their human caretakers across 50 small farms. They used shotgun metagenomic sequencing to comprehensively analyze the antimicrobial resistance genes present in these samples. This approach allowed them to identify and quantify the types and abundance of resistance genes, providing a snapshot of the resistome at the human-animal interface. The study identified an astonishing 270 antimicrobial resistance genes, representing 550 unique types, including critical ones like MCR and BLAUXA58. A striking 52% of these ARGs were shared between humans and pigs, indicating substantial exchange. While pigs showed higher overall AMR abundance, human workers harbored a richer diversity of resistance genes. These shared genes were often found alongside mobile genetic elements, suggesting a high potential for their spread. This research underscores the critical role of the human-animal interface in the spread of antimicrobial resistance, particularly in smallholder farming systems. The identification of clinically important resistance genes shared between humans and pigs presents a clear public health concern. These findings strongly advocate for a One Health approach, emphasizing the need for integrated surveillance and robust antimicrobial stewardship programs to effectively combat AMR. Up next, our sixth paper out of 10, predicting adolescent suicidal tendency in Chinese secondary school students, a machine learning approach with XGBoost and SHAP interpretation by Renjie Yu and Yong Yang from Suzhou Guangji Hospital and affiliated Guangji Hospital of Suzhou University, number 11 Guangqian Road, Suzhou, Jiangsu Province, People's Republic of China. This paper was selected because it addresses the critical and pressing global public health issue of adolescent suicide by developing and validating an interpretable machine learning model, which represents a significant advancement in early detection capabilities. Let's break down its key contributions. Adolescent suicide is a tragic and growing concern worldwide, posing a significant public health challenge. Identifying individuals at risk early on is incredibly difficult because suicidal tendencies are often hidden and influenced by many complex factors. This study aimed to tackle this challenge by developing an interpretable machine learning model to predict suicidal tendency, specifically among Chinese secondary school students. Researchers conducted a large cross-sectional survey involving over 12,000 secondary school students in Suzhou, China. They collected data on 23 different demographic, psychological, and behavioral variables. The team then developed and compared 7 different machine learning models, including XGBoost, to see which could best predict suicidal tendency. The primary goal was to identify students at risk based on these collected factors. The study found that a significant 21.98% of the students exhibited suicidal tendency. Among the models tested, XGBoost emerged as the top performer, achieving an impressive AUC of 0.802, indicating strong predictive power. Key factors driving this prediction included depressed mood, self-dissatisfaction, and a reluctance to seek help. These results highlight the model's effectiveness in identifying at-risk individuals. This research demonstrates that an interpretable XGBoost model can be a powerful tool for identifying adolescents at risk of suicide, potentially enhancing school-based screening programs. It underscores the critical importance of considering both psychological and behavioral factors, particularly for vulnerable groups like distressed males who may not seek help. While promising, it's important to remember this was a cross-sectional study, so future longitudinal research is needed to validate these findings over time and understand causal pathways. Up next, our 7th paper out of 10, Resurrection of Chromosomes from Frozen Animals by Single Chromosome Transfer into Mouse Oocytes by Sayaka Wakayama and Terahiko Wakama from Advanced Biotechnology Center, University of Yamanashi, Kafu, Yamanashi, for 100 Japan. This paper was selected because it presents a groundbreaking method for resurrecting chromosomes from long-frozen animal cells, offering a novel approach to genetic resource recovery and potentially de-extinction that. Let's break down its key contributions. Imagine bringing back genetic material from long-lost species. Traditional cloning methods, while powerful, often fall short when dealing with frozen or degraded samples, as they require intact nuclei and specific oocytes. This limitation has hindered efforts to recover valuable genetic resources from extinct or endangered animals. Our research aimed to overcome these challenges by exploring a novel, chromosome-level revival strategy. Our innovative approach involved taking blood cells from rat carcasses, frozen for over a year, and introducing them into enucleated mouse oocytes. We then used micromanipulation to isolate individual rat chromosomes. Each chromosome was transferred into a fresh mouse oocyte, followed by intracytoplasmic sperm injection using GFP transgenic mouse sperm. This allowed us to culture embryos to the blastocyst stage and generate ES cell lines and chimeric mice. We were thrilled to report significant findings. From our efforts, we successfully generated 17 ES cell lines, with 2 of these lines carrying a full set of mouse chromosomes plus a rat chromosome, totaling 41 chromosomes. Spectral karyotyping confirmed the presence of rat chromosome 9 in these cells. Furthermore, these ES cells generated chimeric mice that displayed GFP-based chimerism across multiple organs, and histological analyses confirmed the expression of numerous genes located on the transferred rat chromosome 9. This study represents a significant leap forward in conservation biology and genetic resource recovery. We've shown that it's possible to functionally revive a single chromosome from a frozen animal, even across species boundaries, opening new avenues beyond traditional cloning. While this is a foundational step, future work will aim towards reconstructing entire genomes and understanding the epigenetic changes that occur during such interspecies transfers. This research holds immense promise for preserving biodiversity and potentially de-extinction efforts. Up next, our 8th paper out of 10, Multi-Omics reveals dysregulation of the Endosome-Lysosome-Autophagy axis and immune-inflammatory imbalance in elderly sepsis by Yi Guo and Dandan Li from Emergency Trauma Center, the first affiliated hospital of Xinjiang Medical University, Urumqi, People's Republic of China. This paper was selected because it addresses the critical and complex challenge of elderly sepsis by employing a comprehensive multi-omics approach to reveal novel, distinct pathophysiological mechanisms, which is crucial for developing precise therapeutic strategies. And let's break down its key contributions. Elderly sepsis, or ES, is a severe condition that combines the challenges of aging with a life-threatening infection. It's known to cause significant illness and death, and critically, it presents with unique biological characteristics compared to sepsis in younger individuals. Our current understanding of these distinct mechanisms is limited, which hinders our ability to develop effective treatments. This study aimed to shed light on these specific pathophysiological pathways. To investigate the unique mechanisms of elderly sepsis, we employed a comprehensive multi-omics approach. We analyzed samples from 4 distinct groups: young healthy individuals, elderly healthy individuals, young patients with sepsis, and elderly patients with sepsis. Our methodology involved untargeted metabolomics and proteomics, followed by integrative analyses and functional enrichment to identify key molecules and pathways. The goal was to pinpoint characteristic metabolites and proteins that could explain the distinct pathophysiology of ES, ultimately proposing underlying mechanisms. Our multi-omics analysis successfully identified a set of characteristic molecules in elderly sepsis. Specifically, we found 2 key metabolites, D-aspartic acid and lyso-PC, along with 7 distinct proteins. These molecules were then linked to several enriched metabolic pathways, including D-amino acid metabolism and glycerophospholipid metabolism. Furthermore, protein-centric functional analyses highlighted terms related to the endocytic vesicle lumen and proton translocating V-type ADPase. Collectively, these findings strongly suggest perturbations in the Endosome-Lysosome-Autophagy axis and an immune-inflammatory imbalance in elderly sepsis. This research offers a crucial theoretical foundation for understanding the complex pathophysiological mechanisms unique to elderly sepsis. The identified dysregulation in the Endosome-Lysosome-Autophagy axis and the immune-inflammatory imbalance represent exciting new avenues for therapeutic development. These findings suggest potential novel targets that could lead to more precise and effective treatments. Moving forward, it will be essential to validate these discoveries in larger studies and translate them into tangible clinical interventions to improve outcomes for elderly patients with sepsis. Up next, our 9th paper out of 10. Nociceptive neurons inhibit neutrophil extracellular trap formation via MLKL-licensed histone release by Han Meng and Yazhou Wang from Department of Neurobiology and Institute of Neurosciences, School of Basic Medicine, the 4th Military Medical University, Xi'an, Shaanxi, PR, China. The Shaanxi Province Key Laboratory of Brain Function Analysis and Modulation, Xi'an, Shaanxi, PR, China. This paper was selected because it uncovers a fundamentally novel and surprising mechanism of neuroimmune interaction, demonstrating that nociceptive neurons can directly regulate neutrophil extracellular trap formation through MLKL-licensed histone. Let's break down its key contributions. Chronic pain is a debilitating condition, and we know that inflammation in the dorsal root ganglia, or DRG, plays a significant role. Specifically, interactions between neurons and immune cells like neutrophils are thought to be key. However, the precise molecular mechanisms driving these interactions and their contribution to chronic pain have remained a mystery. This study aimed to shed light on this critical knowledge gap. To investigate these mechanisms, researchers utilized mouse models, specifically focusing on DRG neurons. They employed various techniques, including MLKL depletion and assays to measure histone H3 binding and neutrophil extracellular trap formation. The core finding was that MLKL in nociceptive neurons plays a crucial role in inhibiting net formation by regulating histone release. They delved into the MLKL histone net cascade, exploring its connection to P2X7 and toll-like receptor 4 pathways. The results were quite striking. They found that peripheral inflammation significantly disrupted the crucial interaction between MLKL and histone H3 in the nucleus of neurons. This disruption led to the release of extracellular histone H3, which then triggered neuronal hyperactivity, net formation, and ultimately, hyperalgesia. Furthermore, when they specifically depleted MLKL in nociceptive neurons, they observed a significant decrease in pain threshold and an exacerbation of net formation. Crucially, interventions like neutralizing extracellular histone or restoring nuclear MLKL successfully reduced both net formation and hyperalgesia, confirming the cascade's importance. This research fundamentally changes our understanding of how nociceptive neurons contribute to neuroinflammation and chronic pain. The discovery of the MLKL histone net cascade provides a brand new and exciting therapeutic target for developing novel pain treatments. While these findings are currently in basic science, they open up promising avenues for future drug development. The next steps will involve exploring how we can translate these intricate molecular mechanisms into effective clinical interventions to alleviate the burden of chronic inflammatory pain. Up next, our 10th paper out of 10. In silico analysis of driver genes in squamous cell carcinoma of the cervix. Insights into their biological functions, prognosis, immune infiltration, and therapy by Samatha Bhatt and Shama Prasida Kubikodu from Department of Biotherapeutics Research, Manipal Academy of Higher Education, Manipal, India. This paper was selected because it addresses a critical global health challenge by comprehensively identifying clinically relevant driver genes in cervical squamous cell carcinoma through the robust integration of 4 independent cohorts and network-based analysis, offering significant insights for prognosis and therapy. Let's break down its key contributions. Cervical cancer is a significant global health concern, being the 4th most common cancer affecting women. Despite its prevalence, our understanding of the specific driver genes in cervical squamous cell carcinoma, SCC, that are clinically relevant has been quite limited. This study aimed to fill that gap by using a comprehensive approach, integrating data from multiple cohorts and advanced network analysis to identify and prioritize these crucial driver hub genes. Our study employed an in silico design, analyzing genomic data from 467 samples across 4 independent cervical SCC cohorts. We utilized a range of computational techniques, including genomic analysis, network-based modeling, functional enrichment, and survival evaluation. The goal was to identify key driver genes and hub genes, and to understand their impact on biological functions, prognosis, and immune infiltration. We identified 749, 109 mutations across 44 driver genes. Notably, PK3CA and TP53 were among the top 10 mutated and hub genes. We also observed significant differences in immune cell composition linked to these hub gene mutations. Importantly, our analysis highlighted 112 clinically relevant compounds, including HER2-directed therapies and PI3K inhibitors, as potential treatments. This research offers a deeper understanding of the genomic drivers in cervical SCC, pointing to specific genes and pathways that could be targeted. While our computational findings are robust, future experimental studies are crucial to validate these driver genes and their therapeutic implications. Ultimately, this work paves the way for developing more effective and personalized treatments for patients with cervical cancer. That's all for today's PubMed Daily. If you found this helpful, please like, leave a comment, and subscribe for daily paper summaries. See you tomorrow.